So this is a Barnacles and the species is Amphibalanus amphitrite. How do you know this? Okay, so because of the stripe and you can see its head, it has a straight operculum, so that's how you know this is a Amphibalanus amphitrite. Now I'm going to dissect this barnacle for DNA isolation from its tissue and hemolymph. I'll show you how to. This video is mostly about how to dissect it in a very rough way, not in a very systematic way. But this thing work also because I have done it so many times. You can do post processing once you get the tissues. You can do the rest of the adjustment. So let's check out. So for the dissection, I took this forceps, which is a thicker one, and I would say key. So you all you have to do is you put one of the forceps end into one of this shell and then break it open from one side only. So how I do is so this is the forceps. So you have to just put this forceps into one of the shell like this and then break open. See, now it's goes completely inside and twist it to break it open. Yeah, now see it's break open already. Now my first job is to take the tissue fluid. So I'll be using a 10 microliter appendrop tube with 10 microliter tips, which is nearby me. So this is how you put the tips, and my first job is to take out the tissue fluid. So as you can see, there are some tissue fluid, it is moving. Now I'll be collecting this tissue fluid in this appendrop tube step by step. Now I'll remove the opercular plate. For that I'm using a pointed forceps. So I'll show you how I do it. See, this is how I remove the. So once you remove one of those opercular plate, its major tissues they come out. All you have to do is use another forceps and just separate the head from it. Okay, and take this tissue into an appendrop tube. For that I'll be putting them into this appendrop tube. Okay, and you can see the major tissue is here. I'll also take the weight of this tissue, but before that let me take out all those hemolymph that is present within the body. Now that I have taken all those hemolymph in a test tube, I wanted to measure the weight of the tissue that I have collected in the test tube, because during any further procedure of extraction of DNA and RNA for gene expression or for protein estimation, you need to take as less as possible the tissue's mass, because the lesser the weight of the tissue, the better contamination, the less contamination and the better volume of your required desired product. So for the video I have shown only the dissection, and maybe in these videos later part I'll show you how to extract the RNA from this tissue that is stored here. You can see something here. So the tissue is here.